Okay, so in this class, we are going to look at a cell, and we're going to look at concepts around the cell. So, what is a cell? So, a cell is just a basic unit of life, or a basic unit of any living organism. That is what a cell is. So, cells are so small that we cannot see them with our eyes, and so we tend to use instruments like the microscope. So we use the microscope that we're able to view the cell. So this microscope helps us to see the cell. Since we can't see it, it magnifies for us the image that we might be able to see it, since we can't see with our naked eyes. So there are two types of microscopes that we usually use when it comes to viewing the cell. So the first one is the light microscope. So the light microscope. So for simple structures, for simple basic structures, we use the light microscope. Then we also have the electron microscope. So electron microscope. So for a more detailed or ultra structure, we use we use this electron microscope. Now, let's look at so on a broader view, when we're talking of cells, they are what we call prokaryotic, prokaryotic, eukaryotic, eukaryotic cells, and also what eukaryotic, eukaryotic cells. So you have prokaryotic and eukaryotic cells. So the prokaryotic cells, these here do not have do not have a nucleus. So these, they don't have a nucleus. So it means that their genetic information or their DNA is just roaming in the cell. Then we have the eukaryotic cells. These have have the nucleus. So in this class today, we're going to look at We're going to look at the eukaryotic cells. So we look at the eukaryotic cells, and these eukaryotic cells, they're basically divided into Into two, which we're going to look at here, which is the animal cell and the plant cell. The animal cell and the plant cell. So what makes these two cells different is just the adaptations that they have. So since one is found in animals, it is expected to have certain adaptations, and another one is found in plants, so it's expected to have certain adaptations that it should be able to be in that plant. So now, so both the animal and the plant cell have certain similarities. So on my left here, you have this animal cell, and then you also have on the other side you have you have the plant cell. So the similarities is that since they are all eukaryotic cells, they have a nucleus. So this black thing here, you're seeing this black circle here is representing a nucleus. That's the nucleus. Then you have you have the cytoplasm. So the cytoplasm, and then you have the cell wall, the cell membrane. I mean, you have the cell membrane. So these three parts: the nucleus, the cytoplasm, the cell membrane. These three here are are similar in both the animal and the plant cell. So now this is when we're looking at at a basic structure or at a simple structure. So meaning when we just view it maybe under under a light microscope. Then the differences is in terms of the structure is that in the animal cell, in the plant cell, you have that plant cells have cell walls, they'll have chloroplast, and they'll have They have a vacuum. So don't worry about this. We are going to We're going to look at each and every part and what that part does. So now we see that since they have similarities and they have the differences. So the similarities, we've seen that in the structure, the similarities are the nucleus, cytoplasm, and and the cell membrane. Then here I have this table. This table shows the differences between the animal cell and and the plant cell. So on this side, here we have the plant cell, and there we have the animal cell. So the differences. So in the plant cell, you have This plant cell has a what? Has a cell wall. And the cell wall is made of what? Is made of cellulose. On the other side, animal cells do not have what? Cell walls. Then another difference is that the plant cell has a large vacuum. So this structure here, you're seeing this structure, this big thing here is a what? Is a vacuum. So they have large permanent vacuums, while animal cells do not have what? Large permanent vacuums. So you see that some animal cells would have small vacuums, but it's not so prominent. So the plant cells are the ones that have what? They have a large permanent vacuum, while animal cells, they do not have. Then plant cells have chloroplast. They have chloroplast, which are these structures here, these structures, and they are structures, those structures are what are usually what? They are green. So the chloroplast structures are what? Are green. They are the ones that give the plant its what? Its green color. So you have this, they are found in what? In the plant cell. Wow, in the animal cell, they do not have what? The chloroplast. Then you see that the plant cells have a regular shape. So you see this here is represented, it looks as a regular shape, which looks rectangular in what? In shape. Wow, an animal cell has an what? Irregular what? Irregular shape. So you just see this shape that is there. It's what? It's irregular. Okay, so now let us look at this on a more detailed on a more detailed scale. So now when we say that when you view the cell using an electron microscope, you will see more parts, or you see more details in the cell. And so now those details are here, and we have that. So on my left here we have the animal cell, and then on the right we have the plant cell. So they'll still have those similar structures, but also will still differ. So like we looked at earlier, you have you have the cell membrane. So in the animal cell here we have the cell membrane, you have the cytoplasm. Then you have now this structure here, which was not there before. That is the rough endoplasmic reticulum. So it's this structure here that has this small round particles, or small round structures here. And these small round structures are called the ribosomes. So these ribosomes, they're even here. So they can either be on the rough endoplasmic reticulum. So they can either be there, or they can be just in the cytoplasm. So the rough endoplasmic reticulum, you also have something similar, which is now the smooth endoplasmic reticulum. What's differentiating the two is just that the rough endoplasmic reticulum has the ribosome on its surface, while the smooth, they do not have. Then you have the mitochondria, which is there, and then you have the Golgi body. Golgi body is here, and all together they are they're referred to as the Golgi apparatus. Then you have the nucleus. So here, all these that you're seeing, nucleus, these just make up the nucleus. So the nucleus, like that. And then when we move to the cell, when we move to the plant cell, you have a cell wall there first, which is the outer part, the outermost part. Then you have a cell membrane. You have your mitochondrion. Then you have your vacuole, and then the vacuole, the membrane of the vacuole is called the tonoplast. That's the membrane of the vacuole. Then you have the Golgi apparatus here. So collectively, we said they are Golgi apparatus, but individually, one like that, you call them Golgi bodies. Then you have the cytoplasm, which is there. We'll explain further what that means. Then you have the ribosomes also. You have the chloroplast. You have a smooth endoplasmic reticulum, the nucleus, and the rough endoplasmic reticulum. Okay, so now let's go step by step, trying to explain what this is. So we'll start with what? We'll start with the nucleus. So this is going to apply for what? So these structures that are in both, they have the function the same way in both of these cells. Either it's in the animal cell or in the plant cell, they will function the same. So the nucleus, this nucleus, we want to understand. We start with the nucleus. So the nucleus, what is its function? So the function of the nucleus is to control all cell activities. The function of the nucleus is to control all cell activities. So everything that happens within the cell is controlled by the nucleus. Another function apart from it, controlling all activities. So all activities, it controls all activities. Then secondly, it stores what? It stores DNA. It stores DNA. So the DNA or genetic information is stored in the nucleus. So these are the two main functions of the nucleus. Then we move on to let's move on to the cell membrane. Cell membrane. So both the nucleus, both the animal cell and the plant cell have a cell membrane. So we see that in the plant cell, after the cell wall comes the cell membrane. Then, since the animal cells do not have cell walls, the first thing that you find there is the cell membrane. That's the outer structure which is there. So the function of this cell membrane. So it is made, first of all, it is made of lipids. So it's made of lipids and proteins. It's just made of Of lipids and proteins. And its function is, it is responsible for what enters and what leaves the cell. So this cell membrane is responsible for what will enter the cell and what will leave the cell. So it is selectively permeable. It is selective permeable. So this means that it selects substances that can go through and substances that cannot go through it. So it selects substances that can go through and those that cannot go through. So you have small substances that it allows, for example, water. So molecules of water can go in, molecules of water can go through it. And then we see that there are big substances that it does not allow to go in or to go out through it. For example, urea. So when you have urea is removed by special carrier proteins out of the cell. So it is selectively permeable. So you must note that selectively permeable. Selectively permeable. So it allows certain substances to go through it and out of it. Okay, so now let's look at the next one. So the next one, we'll look at the cytoplasm. So the cytoplasm, both of them, both in the so you see in here in the animal cell, it's there, and also here in the in the plant cell. So what is the function of this cell, of this cytoplasm? So this cytoplasm is a jelly-like fluid made of of water and dissolved substances. So dissolved substances such as proteins, salts, and sugars. So this is what makes up the cytoplasm. So now this cytoplasm contains organelles or contains cell structures. So these cell structures that you are seeing, the mitochondria, the vacuole, all these here which we are looking at, the ribosomes, all these are the organelles or the cell structures, and they're suspended in the cytoplasm. So this is where this is the site for cell activities, where these activities of the cell happen. So this is what about the cytoplasm. Now, fun fact that you must know is that when we collect, so the combination of the nucleus, the cytoplasm, and the cell wall, and the cell membrane, and the cell membrane, all together, collectively, these three are known as the protoplasm. Protoplasm. So these three collectively, they are called protoplasm. Okay, let's now move on to the next one. So the next structure, or the next part that we are looking at is this here, which is the mitochondria. So here it's there, the mitochondria, mitochondrion, like that. So mitochondrion, that's a single one. Plural, it's going to be mitochondria. This side on the plant cell, you have it there. That's the mitochondria. So this mitochondria, also known as the powerhouse of the what of the cell. This is just a rod-like structure or a sausage-like structure where respiration what occurs. So its function is to what, to house respiration. This is where respiration what takes place. So that's the function of what, the mitochondria. Next, we go to the ribosomes. So these black round structures you're seeing here are called the ribosomes. And the ribosomes, the function of the ribosomes here, the function of the ribosomes, this is just the site of what, of protein synthesis. So in short, that's where proteins are made. Proteins in the cell are made in the what, in the ribosomes. The next structure we have is the endoplasmic what, reticulum. The endoplasmic reticulum. And of course, we have two. You have the smooth endoplasmic reticulum and the rough endoplasmic reticulum. So in general, the endoplasmic reticulum is a network of membranes. A network of membranes used for transportation of substances within the cytoplasm. So it's just a network of membranes used for transportation of substances within the cytoplasm. So now, as we said, you have the rough. So you see here the rough endoplasmic reticulum. So the rough endoplasmic reticulum has the ribosomes on what? On its surface. That's the rough endoplasmic reticulum. And its function is to transport proteins. Its function is just to transport proteins. Then you have the smooth endoplasmic reticulum, this one here, which does not have the ribosomes on its surface. And its function is just to transport lipids. Its function is to transport lipids. Okay, we move on to the next structure. The next structure is the Golgi bodies. So the Golgi bodies, or collectively, as we say, the Golgi what? The Golgi apparatus. So these here, they carry proteins such as enzymes from the site of synthesis. So from where they were made to the site of what, of reaction, to where they are going to what, they are going to be used. So this is what the function of the Golgi apparatus, or the Golgi what, the Golgi bodies. Then now let's go on to explain the last three structures here, which we have not explained. That is the ones that are just in the what, in the plant cell, and that is the chloroplast. So the chloroplast, these structures here, these here, the chloroplast, this here, this here, this, just the chloroplast. So the chloroplast, this, their function is what, is to carry out photosynthesis. So these are structures, green-like structures, that in fact give the plant its green color. So the function is just what, to trap light, and that light is used for what, for photosynthesis. Okay, so which is just the plant making what, its own food. Then you have this big structure here, which is the vacuum. So this vacuum is a fluid filled space inside the what, inside the cytoplasm. And it contains what, inside this vacuum here, it contains cell sap. It contains cell sap. So cell sap plays a very important role. So the cell sap, what's just inside the cell sap? The cell sap is just a solution that contains salts. It contains salts, sugars, and what, and water. That's the cell sap. contains salts, sugars, and water. So now, the concentration of the cell sap plays a role in the what? In the movement of water into the cell and out of the cell. So the concentration of the cell sap plays an important role of water going either inside the cell or moving what? Outside the cell. So now we'll get to understand in further lessons that uh, if the inside is what is concentrated more than the what than the outside, then water is going to go inside the what the cell. But if the concentration outside the cell is greater than that inside the cell, then this vacuum here is going to what to lose water like that. So it helps to balance the cell sap, helps to balance the amount of water that is in the cell and outside the what the cell. Then the last structure we're looking at is the cell wall. The cell wall. So this cell wall is the outermost boundary of the what? Of the plant cell. And it is made up of what? Of a substance called cellulose. It's made up of a substance called cellulose. Cellulose, like that. Okay. So now it is important. So it's important because it protects against damage, and it prevents what? It prevents bursting when the plant gains a lot of what? A lot of water. So when water goes into the what? Into the vacuum swells and it begins to push against the cytoplasm, and it will continue to swell like that. Now, because the cell wall is made of a tough structure, which is the cellulose, it will prevent the cell from bursting. Instead, it will just become turgid. And then you have, because of that cell wall, the cell wall will give this plant cell a regular structure. This cell wall itself is what? Is fully permeable. Fully permeable means it allows anything to go in. It does not select anything. Anything, it goes through it. It can go through it. But then there's a cell membrane which will then select what will continue to go in, and that which will not go in. Okay, so this is just about the parts of the cell, and then even their functions. So let's look at a past paper question on this cell. So you have this question here. It shows a plant cell. So they have told us that figure 1.0 shows a plant cell. And then here they're telling us now that there's part A, part B, and part C. Part A, part B, and part C. So the next, or the question A here says, name the parts labeled A, B, and C. Names the part labeled A, B, and C. So A here is this big part here. And we see from what we just from learning, the biggest part of the plant cell there, or structure, was the vacuum. So this here is going to be the vacuum. So you write vacuum there. And then B here is this black spot there, or black thing here, or black structure there. So that there, as we saw in the previous diagram which we looked at, that's just the nucleus. That's the nucleus. Then you see C here is pointing at the outermost structure of this cell. And since it's a plant cell, the outermost structure is is the cell wall. So C is going to be cell wall. Then part 2 says, name the organ in the plant in which the figure is found, given and where the figure is found. Then they're saying give a reason for your answer. So the organ where we can get this plant cell, or where this plant cell was found, is the leaf. Is the leaf. And the reason why is because it is showing this. You see that there are a lot, or the presence of a lot of of these structures that are that are colored here, that are a lot in the cell, and those structures there are the chloroplast. And the leaf has most of the chloroplast, that it's able to trap a lot of light for the cell to use for photosynthesis. So because of the presence of the many chloroplast structures there, which of course trap light, which is used for photosynthesis. So that's the reason in your you have there. Okay, then part B is saying describe two adaptations of the cell in figure 1.0, which enables it to carry out its function. So two adaptations of this cell which helps it to carry out its function. So the first adaptation is, we're going to say, it has a large vacuum. So as you see, this part A here is a vacuum. So large vacuum. The presence of a large vacuum. So that large vacuum helps it to maintain temperature, pressure, helping the cell to stay firm. So it helps it to be turgid, that it should stay firm and in place. So that's one adaptation. Then the other adaptation is that you see that the outer structure is the cell wall. So it has a thick cell wall. So thick cell wall. So the thick cell wall provides a structure for support and protects the cell from damage, as we earlier learned. Okay, then the last question is saying, explain what would happen to the cell in figure 1.0 above if it was placed in a test tube containing distilled water. So if you place this cell in distilled water, what would happen? So the cell would take in the water by osmosis. And that is because this vacuum here, the vacuum, which is filled with a cell with a cell sap, as we said, it has salts, it has sugars and all. So since it's been placed in distilled water, it means that the concentration that is inside the cell is higher than that which is outside the cell. So meaning that water is going to move from a region of lower concentration to a region of higher concentration, which is just osmosis. So that is the process of osmosis. So if it's placed in distilled water, osmosis is going to take place, causing this vacuum here to swell. So the vacuum is going to swell and it's going to become turgid. Now, because there's a presence of the cell wall, it will not burst. So it will keep it just turgid like that. So that is what would happen if this cell is placed in distilled water. Okay, if you're new to this channel, remember to hit the subscribe button, and we will see you in the next one.